Okay, Chapter 4 is a tour of the cell, and we're going to start by looking at how living things have changed the environment. Often we look at how life responds to the environment, but we're going to start by looking at prokaryotic cells that have literally changed the face of the earth. These ancient bacteria have been around for billions of years, and they have been oxygen for about a billion years. Didn't make it really into the atmosphere. It got used up, and for about a billion years, the earth simply rusted. The iron in the soil had not been exposed to oxygen until then, and when it was exposed, it rusted. It turned red. You can see it in the soil. And after that reaction was complete, then the oxygen could start being free in the atmosphere, and more advanced forms of life need oxygen. So the Earth formed about 4.6 billion years ago, and the early atmosphere didn't have oxygen. It had water, carbon monoxide, which is poisonous, carbon dioxide, nitrogen, which it still has today, and methane. We think it was pretty violent. Volcanic activity, lightning, and UV radiation were intense because right now we're protected from the UV radiation by ozone, which is a form of oxygen, but it wasn't there yet. So, how did life originate? You have organic molecules, and we think it may have been a physical process, meaning abiotic. It's not a life process. We think it was physical formation of these organic molecules from the molecules in the environment. And exactly how that's done, there's been some experiments that show that you can take what was present in the early atmosphere and you can make polymers and organic compounds essential for life. Stanley Miller is the one who showed that this could be done, and basically what he took was he made a contraption, and he took right here you can see the early ocean is used in his simulation, and then for the atmosphere he had the methane, the nitrites, the hydrogen, and then an electrode right here to simulate lightning. And what he was trying to do was to make these organic molecules right here, to get organic molecules, the building blocks of life. And sure enough, he could create that. The first polymers were probably formed on hot rocks or clay near the ocean, right? You see these things right in the tidal areas. And a polymer, remember, is just you take a monomer, a single unit, and you link it together and you get a polymer. So you can make polymers of amino acids and you can get proteins, or you can take nucleotides and create nucleic acids. Probably RNA for the first nucleic acid. That first genetic material was probably RNA, and the reason for that is that it, right here, it can catalyze their own replication. That's huge. DNA needs proteins. It needs stuff to catalyze it. RNA can do it itself. It can self-replicate, or it can be used to make proteins. It can do that both. So the first genetic material was most likely RNA. Okay. Membranes can be created, and so you can get these membranes enclosed cooperatives, which is essentially the beginning of a cell, but it's not a single unit yet. A cooperative just means there's stuff that happens to be enclosed in this membrane, and that stuff would be RNA is the key. Okay, and then RNA, remember, it can self-replicate, but it can also be used to create polypeptides, the beginning of your proteins. So not only can it make its own RNA, it can also be used to make polypeptides. Okay. Your membranes probably were able to separate these aggregates, and in this picture you're going to see you have a membrane inside of a membrane, and that's kind of the beginning of a cell. when you can start separating things out. So you can separate out your RNA and your polypeptides. Ultimately you're going to get a nucleus. You're going to get your genetic material enclosed in its own membrane. And that would give these molecules this advantage of being protected from the environment. Okay, prokaryotes have inhabited the earth for billions of years, right? It's the oldest life form. They're the ones that created the oxygen, and really they had to come first to create an environment conducive really to other kinds of life. They are still pretty much everywhere. They are all over Earth. They go all the way down to these deep sea vents. They are ubiquitous on the planet. They're very well adapted to live here, and they reproduce quickly. Okay, bacteria and archaea are the two main branches of prokaryotic evolution, and the way that you can tell them apart is through their nucleotide sequences. They diverged probably billions of years ago, and so maybe not to the naked eye do they look different, but their genetics are quite different for the two different branches. Okay, prokaryotes come in a variety of shapes. You have the cocci, which are these little circular ones right here. They're going to be like little balls. All right, bacilli look like little rods right here. And then they can also look like little curves or spirals over here. External structures of your bacteria can be a cell wall, and a lot of them will have a capsule. They can be covered in a sticky capsule, and this will protect the bacteria from the environment. It'll just protect everything in it. It also kind of makes it hard to kill bacteria. Some prokaryotes have pili, and what pili do is they can stick it to a substrate. And in fact, anyone who has those sonic hair toothbrushes, one hypothesis as to why they work so well to prevent plaque and stuff is the bacteria in your mouth have pili. They actually have flagella as well to move around. And they think that the vibrations in the sonic hair toothbrush just rip the pili right off the bacteria, and then the brush or your mouth can just kind of get rid of the bacteria. But when it's stuck to your teeth, it's a lot harder to get rid of. Okay. A lot of bacteria and archaea have these flagella, and that enables them to move. So down here you'll see you literally get like this thing's going to spin, right? This little flagella is going to spin, and that will make the bacteria move off to the left. In this case, it will move away from the flagella. And that allows, it allows them more access to the environment, and is another adaptation for survival. Okay, one issue with prokaryotes bacteria is they reproduce really quickly in favorable environments. So if you get sick with a bacterial infection, one problem is that you would be considered a favorable environment for that bacteria, and it's going to reproduce really, really quickly. So when you take your antibiotics, usually you have to take it, 10 days to make sure you've killed all of them, because even if there's just a few, then they will just reproduce again very quickly. It can be hard to get rid of bacterial infections just because of their reproductive rate. Okay, some prokaryotes can withstand harsh conditions. So when the environment's favorable, they reproduce very, very quickly. When the environment is not favorable, they will form an endosperm, which is a protective capsule which will keep all their RNA, all their genetic information safe from the environment, and enough that when the environment then does become favorable, they can go back and do these metabolic processes, including reproduction. So the endospore is sort of a form of stasis. It's not going to be reproducing, but it'll just stay in these harsh environments waiting. Okay, prokaryotes obtain nourishment in a variety of ways, and they exhibit much more nutritional diversity than eukaryotes. So what does that mean? Well, we have to eat. you and I, we have to eat things for our nourishment. Prokaryotes can do photosynthesis. They can use chemicals to make their nourishment and to get energy from chemicals. They can do a variety of things that we don't do. Okay, autotroph. Autotroph means self-feeder, and so it's going to be an organism that feeds itself. Well, how does it feed itself? A photoautotroph would make its organic material from the energy from sunlight, that's the photo part, and carbon dioxide for the carbon, and it can use that energy to build its own building blocks for what it needs. A chemoautotroph is going to obtain its energy from inorganic chemicals. Most likely you'll find these in the deep sea vents where you have lots of harsh chemicals. Heterotrophs, on the other hand, are other feeders. They have to eat something else. They obtain their carbon atoms from essentially eating it. A photoheterotroph will, and we don't have these when we think of plants, they're photoautotrophs, but a photoheterotroph will obtain its carbon from the environment, but get the energy to make that carbon into something from sunlight. And a chemoheterotroph will obtain the energy from chemicals, but it'll obtain the carbon from organic compounds, from eating something else. Okay, you have specialized membranes that perform metabolic functions. So what that means over here on this left one is if this is a respiratory membrane, then this is going to be a heterotroph. It's going to use metabolism to create it's going to eat something and use the metabolism to create its energy. On the right side, you're going to see a thylakoid membrane. So this is going to be your photoheterotroph or photoautotroph. This thylakoid membrane will be used for the photosynthesis part of it. Archaea thrive in extreme environments, and what you will see here on the right is this is Yellowstone out here, and this is one of those crazy hot, sulfury, nasty smelling lakes out there, and you can even see some dead trees around it. Really, really harsh environment. And on the left, this over here, this is not dirt. This red is actually the Archaea right there, and that will be in extremely salty conditions. They will thrive. They are perfectly happy in things much too salty for us.